Hi friends, this is Priyanka. Welcome to Entomology Coaching. Before the session, I kindly request you guys to watch my video on a collection of soil and root samples for nematode extraction. This session is exclusively for the nematode extraction methods. But before get into the extraction procedures, I kindly request to note these points. Regarding the sampling methods, we can undergo a field crops, vegetable crops, fruit crops. The field crops at the depth of 15 to 20 centimeter and vegetable crops at the depth of 20 to 30 centimeter and fruit crops at the depth of 30 to 60 centimeter. Regarding the field crops and vegetable crops, we have to use a shovel for the experiment. Shovel, an instrument for the experiment, and the fruit crops for auger, AUGER. And next, the nematode is stored at 10 degrees Celsius for 2 weeks if when immediate extraction is not done. Depending upon the nematode feeding habitat, we have to collect the sample from different places. Like if the nematode is of ectoparasitic in nature, we have to collect the samples from soil. If it is of semi-endoparasitic in nature, endo semi-endoparasitic in nature, we have to collect the nematode along with soil and plant root. If it is an migratory endoparasitic, we have to collect it from a plant and a soil. If it is a sedentary endoparasite, we have to collect it from plant alone. If it is an Aphelenchoides and Anguina, we have to collect it from seeds since they are seed-borne disease. Next, we talk about extraction methods. Four important extraction methods are there. One is corpse decanting and sieving method, Behrman funnel method, modified Behrman funnel method, and last one is sugar flotation or centrifugal flotation method. Apart from this, two more methods are there. That is Eastern Brink method, mainly based on the principle of elutriation. Elutriation is counterflow of water. And another method is Steinhorst Erlenmeyer flask method. It is a two flask method. We can see less about these two methods, and we can see about four methods in detail. Let's get into the corpse decanting and sieving method. Okay, regarding corpse decanting and sieving method, we can see about their principle is based on the differences in the specific gravity. The soil particle and nematodes get settled at the different rates. First, we have to take a pan 1. Thoroughly mix the composite soil sample. Take a soil sample of about 200 cc. 200 cc in a pan 1. In pan 1. Next, add about 1 liter water and stir well and break clots and clumps. Wait for 4 to 5 minutes and pass this soil suspension to pan 2. Soil suspension to pan 2 with a soil suspension mixture. Okay. Then repeat this process 2 more times to ensure that no nematode is left in pan 1 and discard the remaining material. Stir the suspension of pan 2 gently and wait for 4 to 5 minutes and pour it through the series of 60, 100, 200 and 350 mesh sizes. That is, mesh is nothing but a number of apertures. Apertures is holes, a number of holes per linear inch. Let's talk about some more about mesh. Mesh is number of apertures. number of apertures per linear inch. Per linear inch. Most important question, friends. Next, it's a mesh number and pore aperture in micron. A mesh number and pore aperture in micron. If the mesh number is 20, 840 number of holes in micron is there. If it is 60, it is of about 250. Like that, if it is 100 mesh number means they have a number of aperture is 150. If it is 200, it is 75. If it is 350, it is 45. So more the number, less the apertures. It is exactly reciprocal. Okay guys. After collect the residues left over 60, 100, 200 mesh sizes separately in the beaker and label it. Sixty mesh size is important for the cyst. 100 for large size nematode. 325 for juveniles. 400 for X. Okay guys, let's talk about Behrman funnel method. Regarding the Behrman funnel method, active and a motile nematode tend to move towards water through the tissue paper and gets collected at the base due to the gravitational force, whereas the inert soil particle and a debris remain on the tissue paper. Let's get into detail in it. Procedure. First procedure is similar as like a cup sieving and decanting method. Second thing is arrange the assembly which contain arrange and assembly a stand which contain a glass funnel along with a rubber tube with a vial below to it. This clip is Hoffman's clip. This clip is Hoffman's 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 clip. And about to the glass funnel a wire mesh is there and a double layered filter paper is also placed. Before that we have to fill it with full of water into the funnel. Next thing is active and a motile nematode can be collected when it touches the tissue paper with water. Advantages is there. The clear nematode suspension without debris can be obtained and the nematode collected in a small amount of water. Disadvantages is that a more time consuming. A nematode can lose their activity due to lack of oxygen. Sometimes it may occur. And regarding the sedentary and slow moving nematode cannot be extracted using these methods. Next one is Behrman with a modified type, that is modified Behrman funnel method. This is exactly same as to the Behrman funnel method, but in case of funnel we take a petri plate upon it. A petri plate above which a wire mesh is placed and above to is a filter paper is placed. And next method are similar to the before one. And the next method is a centrifugal flotation method. This principle is a difference in a specific gravity of the nematode and sugar solution results in a separation of nematode using a centrifugation method. See, in this centrifugal flotation method, centrifugal flotation method, a difference in specific gravity of the nematode and a sugar solution plays an important role. For the sugar solution, the specific gravity is 1.18, and for the nematode, the specific gravity is 1.05. 1.05. So this differences results in separation of nematode using centrifugal method. Next one is their procedure. The process the soil sample by corpse decanting and sieving technique and collect the suspension in a beaker. Distribute the suspension into the centrifugal tubes equally and add water if necessary. And centrifuge at 3500 rpm. 3500 rpm for 3 minutes. Discard the supernatant solution and retain the soil residues containing nematode. Then fill the half of the tubes with sugar solution. See, this is a tube with a soil residue containing a soil residues containing a nematode. Now fill the half of the nematode with a sugar solution and shake it well. Then fill the sugar solution from 0.5 centimeter from the top. 0.5 centimeter from the top, again fill it with the sugar solution. Next, centrifuge at 3500 rpm for 1 minute and decant the supernatant suspension from each tube and pass to 350 ml sieve. Never leave the nematode in a sugar solution for the longer time and wash the residues on the sieves repeatedly with plain water to remove the sugar solution. Transfer the residues on the sieve that containing nematode to a beaker for observation. That's all, friends. Thank you.